Okay friends, so welcome again. So as we have understood the concept of colony hybridization, plaque hybridization and colony PCR very well, now it's time to solve a numericals on the basis of this. Okay, so let's start. So first of all, as usual, we will read the question very carefully. So start. Which of the following partial amino acid sequences from a protein whose gene you use to clone would be most useful in designing an oligonucleotide probe to screen a cDNA library? Okay. Again, so which of the following partial amino acid sequence from a protein whose gene you wish to clone would be most useful in designing an oligonucleotide probe to screen a cDNA library? That means what they are telling us. Let's try to analyze. That means it is a protein, and this protein is coming from a gene whose of which we want to clone, right? Now they have provided two kind of protein sequence. That protein sequence, when if we perform a reverse translation, then reverse transcription, we will get two DNA sequence. Okay, that means what? These two sequence are nothing but some portion of the total DNA sequence which ultimately coding the total protein, right? Now they are telling us that on the basis of which DNA sequence you will make your probe. Okay, that means what? During the explanation of colony PCR, I have told you the first choice of you will be to find out a unique sequence of gene of interest. Okay, so they are want to know from us that according to you, what gene coming from those kind of peptide sequence will be a unique, that will generate a unique sequence for the entire DNA from which the entire protein is coming. Okay, so that is the idea. So for that you have to know about the basic of transcription and translation. Okay, so let's try to understand it at a very basic level, that we have a double stranded DNA molecule. Okay, so this double stranded DNA molecule will replicate to increase its number, and it can also undergo translation to produce mRNA, and from the mRNA, that is the positive sense RNA, it will convert it into protein, right? And how it is converting? It is converting with the help of two player. One is ribosome, another one is tRNA. Okay, when we will launch our molecular biology lectures, we will going to learn about this in details. Okay, so now this ribosome will go through the mRNA and generate the peptide chain, okay, with the help of tRNA. Okay, so this tRNA contains anticodons, and remember, in mRNA the three nucleotides together collectively called codons. Okay, so suppose it is a mRNA chain, so the tRNA will bind over here, and it contains a three nucleotide, that is here anticodon. On the basis of that anticodon and codon reaction, the particular amino acid that it is carrying will be inserted here. That is the idea of normal translation. Okay, so that means the amino acid methionine is coming from 3 nucleotide, amino acid leucine is coming from 3 nucleotide, amino acid arginine is again coming from 3 nucleotide, and leucine is also similarly those things also. Okay, so these 3 nucleotides are collectively called codons. Okay, so here I want to discuss with you about two important properties of codon, and I will also give you a mnemonic. Okay, that is quite funny, but don't take it seriously. This is just to remember it. Okay, so Remember, I want to told you about the codon degeneracy and codon universality or universal codon or an unambiguousness. That is the unambiguousness of the codon. Okay, that the codon is universal and unambiguous. I hope you have learnt about unambiguous nature of the codon and the universal nature of the codon and the codon degeneracy. So today I am going to give you a trick. Before going to do this, I want to told you that the trick I am going to provide you, it is just for fun. Okay, so that you can remember it very well. Okay, so and don't take it seriously, please. So I have told you that 3 nucleotide sequence collectively are called codon, and this codon via anticodon reaction facilitates the attachment of amino acid, right? So I want to mention these codons as boyfriends. Okay, and those amino acids as girlfriends. Okay, now I want to told you that there can be possibility that amino acids can have multiple codons, and that is called obvious, right? But the codons are universal, they are committed towards only one girl. Okay, that means I want to just tell you about that codon is specific for a particular amino acid, but amino acid may not be specific for a particular amino acid. Okay, that is the codon degeneracy. Okay, and codon universal and codon unambiguous nature. Again I am repeating that amino acid can have multiple codons. Okay, that means the suppose our leucine have 6 different codons. Okay, that means leucine have 6 different boyfriends, but each of those boyfriend consider leucine as their girlfriend. That means codons are universal and unambiguous, and this phenomenon that an amino acid can be recognized by multiple type of codons is known as codon degeneracy. Okay, after that I think you cannot forget this anymore. Okay, so on the basis of that what codon is specific for what amino acid, there is a codon chart that I want to provide you that will help you to understand the question more specifically. So this is the codon chart that simply saying us that for phenylalanine there are 2 codons. Okay, for leucine there are 2 codons and again 4 codons, that means in total 6 codon. There is 3 codon for isoleucine, methionine, valine. That's how it is going on. So when we will going to discuss about the codon charts during molecular biology lectures, I will surely discuss with you in details. So in that question we have to find out which of the amino acids have minimal number of codons. That means what? Minimal number of codon means specificity will be higher, right? But maximum number of codon means if suppose we will choose leucine, arginine as our probe, that means they can bind with anyone, right? Because here 2, 4, 6 and again that means total 12 different type of codon, right? That means it cannot provide you specificity. But if you consider methionine containing protein and tryptophan containing protein along with let's say histidine containing protein, then that particular amino acid chain will develop a DNA via reverse translation, and reverse transcription will be specific, right, as compared to the first case. That is the question. Okay, So here I have mentioned that methionine has only one codon, and I hope you all know about this, that is AUG. Okay, leucine have 6 codon, arginine has 6 codon, and leucine has 6 codon. That means 6, 6, 12, 13, total 13. That means the specificity will be significantly lower, right? On the other hand, methionine has 1 codon again, tryptophan has 1 codon, cysteine has 2, and tryptophan has 1. That means 1, 2, 3, 4, total 4. That means now you tell me which one provide more specificity. Obviously the Q, that is the idea. Okay, so Q will be the more useful. It will ensure uniqueness rather than the P. Okay, so that is the question that we have to solve. Okay, they are just providing you two amino acid sequences, and they want from you that which amino acid sequence you will consider to screen the particular gene that will code for the particular protein. Okay, so that was the question. So I hope this video will be helpful to you, and I would like to apologize for using this kind of funny tricks. Okay, so thank you for listening to this class.